Hi, everybody. My name is Liang Huang from Oregon State University in BioResearch USA. Today, I'm going to present to you our work on linear design, which is an algorithm to design the best, most stable, and optimized mRNA sequence for vaccines and mRNA medicine, which improves stability and immunogenicity. And this is a work which is currently under revision at Nature. And as you all know, mRNA vaccines have been great to combat this current pandemic, but it has a catch that it suffers from chemical instability because mRNA vaccines, mRNA molecules are mostly single-stranded and they easily degrade. And once they degrade, they cannot produce the spike proteins that you wanted. And as a result, they trigger less antibody response or less protection. So our goal in this work is to design more stable and efficient mRNA sequences so that you don't need to use it for cold chain storage or transportation, and you can use less dosage and it will induce less side effects, hopefully. So the problem of mRNA design is very simple. So it's basically the inverse problem of protein translation. And in protein translation, it's like a deterministic process, but in mRNA design, it's non-deterministic because of the redundancy in genetic code. Each amino acid, like valine or leucine, has multiple choices of triplet codons. Like valine has 4 choices, leucine has 6 choices. And if you multiply all the choices together, you got a humongous number. That's basically a combinatorial explosion. Exponentially many possible mRNA sequences. And they're all valid. They're all synonymous. They all translate to the spike proteins. But which one should you use? Well, Moderna's paper right before the pandemic suggested that you should choose the most stable mRNA because that would produce the most amount of the proteins. But the question is really, how do you choose over this humongous number of candidates, the most stable one? Well, you cannot enumerate them one by one, so that would take billions of years. So you borrow ideas from natural language processing and which reduce it to a kind of a problem solved 60 years ago before the pandemic, which was called lattice parsing, which I will not go into any details. I will just show you the results. Here's the results. With about just 10 minutes of optimization, this is so efficient, we can design a completely different sequence for the spike protein, which of course both of them translate to spike protein, but this wild type is so unstable. As you can see, you have tons of single-stranded regions like these red parts, which can be easily cut down into pieces. But our design is so stable, it's mostly double-stranded. It has mostly paired and has the lowest free energy that you can prove. Now, a vaccine company in China, Stemmer Therapeutics, did wet lab experiments for us, all the way to animal experiments. And they showed using design similar to this design, you can induce up to 23-fold increase in antibody response compared to conventional codon optimization designs like those used in Moderna and BioNTech and Pfizer. And this is not just stability. We can also optimize jointly with codon optimality, which is CAI, codon efficiency or translation efficiency. So on the x-axis, you have the free energy, which is the stability, the lower the better. And on the y-axis, you have the codon optimality, the higher the better. But traditional technique of codon optimization only optimizes codon optimality, like those used by Moderna and BioNTech and CureVac, and including our baseline. But actually, this is a two-dimensional space, and nobody realized that there's a huge territory which is completely uncharted beyond this line. That is the more stable ones. But there is kind of a feasibility limit. That's the blue curve here, which is beyond that curve, there's no design that is available. But lower right of that curve, everything is possible, and you have a huge number of possible candidates. So this is huge uncharted territory that waiting for us to explore, and we explored some of them, which showed up to 23-fold increase in antibody response. But actually, there are way more candidates in here that we haven't got a chance to explore. So possibly somewhere here is even better. So this technique is currently being used by a lot of vaccine companies all over the world, including Sanofi, which is one of the largest pharmaceuticals, who have licensed it with milestone payments upon entering clinical trials. And this paper has received positive reviews from Nature and is currently under revision. And this is more than mRNA vaccine, for sure. You can design any mRNA sequences for any therapeutic proteins. You can use our algorithm to design mRNAs for monoclonal antibodies, anti-cancer drugs, CAR T therapies, you name it. And this is going to be the future of personalized medicine, because we'll just give you the prescription, which is the mRNA sequence, and your body, your cells will be responsible for basically translate them into the protein drugs that you need. So your body is your own manufacturer of your own drugs. And with that, I would like to thank you for your attention, and please take a look at our paper preprint and trial web server. Thank you very much.